All right, so how to make a protein. We can only start this, and hopefully with tomorrow's class we'll go a little farther and we'll put it together. All right, so hey, we need proteins. We use proteins for everything. Enzymes, you already know the mouse for the epistasis example. Everybody good with that, right? Yes. In the 1900s, I'm talking like 1910, 1920, people knew that genes were needed to make enzymes. If you had the wrong enzymes, you got diseases. Both of those things were correct, but at the time they were just ideas. We now know that is true. They were absolutely correct. Hey, 3 differences between DNA and RNA. They were in your lab already. What are they? The difference in what? Always.
The structure is not right.
Well, I'm looking for, yes, structure is right. Go ahead and say it.
The uracil.
Yeah. What base? You're right. Uracil versus thymine,
Yeah.
okay? So there's always a difference in bases. The DNA for the base is going to use thymine where the RNA would use uracil. They both use adenine, cytosine, guanine, but DNA uses thymine, RNA uses uracil. That's a difference in the bases. There's another difference, it's in their names.
Sugar.
Yes, what sugar they use. DNA uses deoxyribose and RNA uses ribose. And Brooke, no. Natalie, and what is the usually true about DNA versus RNA? Yes, DNA usually 2 strands, RNA usually 1 strand. And Brooke, what's an exception to that? What is a 2-stranded RNA in humans?
The siRNA.
siRNA, very good. All right, let's move on. So, you know that if we look at RNA, just like DNA, it's made out of nucleotides, made out of a sugar, a phosphate, a base, but this time the sugar is ribose. Everybody good with that? You know that we have adenine, cytosine, guanine, and uracil, right? Okay. It's pretty straightforward. So RNA is still made out of nucleotides just like DNA. There are different types of RNA that I want you to know. I want you to know messenger RNA, ribosomal RNA, transfer RNA. What do all those have in common? They're all what? All single strand. Okay. A ribozyme acts like an enzyme, meaning it folds up into a cool 3D shape. It catalyzes the reactions, speeds them up, lowering the activation energy. siRNA is small interfering RNA. We'll live, it's okay. Besides, we can edit. All right, siRNA is a small interfering. That one is double-stranded, and it's in red. Let's move on. Okay, Bryson, you're on candid camera. Okay, so you understand the gene expression. What happens if we read the gene? If we haven't, we're not reading it. It's just part of the genotype. But if we read it, if we express it, it could become part of the phenotype, right? Okay. So, proteins are a chain of amino acids, and their shape is controlled by what amino acids we are using. Everybody good with that? All right. Now, in transcription and translation, this is related to what we did in lab last week. You're going to use 3 types of messenger RNA, ones I said I wanted you to know. 3 types of RNA, the ones I said I wanted you to know. Messenger RNA is, in that cartoon, the recipe, right? It is the instructions how to make a protein. Now remember, when we first read the DNA and copy it into RNA, we're going to have to remove the introns before we can use it. We hook the exons together. The exons hook together, and only the exons are what are going to be the messenger RNA, the message that goes out of the nucleus. The ribosomal RNA is the main part of a ribosome. You remember I said a ribosome is like a chef? You have the human, okay? But they're not a chef till they wear that little poofy hat. Same thing. The ribosome is mostly the big subunit, rRNA, but it has a little cap of protein. Until it wears a little cap of protein on top, it's not a full ribosome. Good? All right. So transfer RNA, that's our sous chef, that's our helper chef. It brings amino acids to the ribosome. Everybody good with that? Yeah. So no issues there. The amino acids are the ingredients in the protein we're going to make. Genes are written in codons. So we have a group of 3 nucleotides, that's a codon. A codon of messenger RNA tells us what amino acid we're going to use. Good? You've seen the chart already. The ribosome is going to read the messenger RNA, and it's going to look for a transfer RNA that matches. So the transfer RNAs are bringing amino acids, and the ribosome is looking and say, hey, do you match? The reason it can do that is because there's a piece on the transfer RNA that tells it, yeah, I match there's a piece on the messenger RNA called the codon. There's a piece on the transfer RNA called an anticodon. If the anticodon matches the codon, you must have the right amino acid. Good? All right. So you're all familiar with this. You know that transcription happens in the nucleus. It's reading the DNA, copying the information to RNA. You know that translation happens outside the nucleus. That's reading the messenger RNA to make the protein. You know that transcription is done by RNA polymerase. You know that translation is done by a ribosome. Everybody's good with that, right? That's review, you've heard it? All right. So RNA polymerase, in our cartoon, the monk-like scribe, it's an enzyme that reads the DNA, makes the RNA. It reads a little part of the DNA, one gene. It starts at the, look for a P word. Yes, it starts at the promoter and it stops at the terminator. Yeah. You start reading DNA in the promoter, you stop reading in the terminator. Good. Okay, so RNA polymerase will bind to the promoter. It will unwind and unzip the DNA all by itself. This is important. This is a test question. I want you to know that RNA polymerase needs a lot of help. It needs other proteins to unwind the DNA, other proteins to unzip the DNA, other proteins to hold the 2 strands apart. Okay? Sorry, I should have said DNA polymerase needs a lot of help. Okay? DNA polymerase needs other proteins to unwind the DNA to hold the 2 strands apart. RNA polymerase is fine. RNA polymerase does the whole job itself. It reads the DNA, it unwinds it, unzips it, it copies it, it puts it back together. Are we clear? Okay. So know that RNA polymerase is smarter than DNA polymerase. It does all the stuff by itself. Doesn't need helicase, topoisomerase, binding proteins. Okay. Transcription is reading the DNA, making RNA. You're going to start on the promoter of the template strand and read until you get to the terminator of the template strand. You don't even use the other strand. Why is it there? It's a backup copy so you can fix mistakes in the DNA. Does that make sense? Okay. Oh wow, you've seen a table like that before. You need to know what transcription is, you know what translation is. You know that when we're doing transcription, we start at promoters and stop at terminators. You know that when we do translation, we start at start codons and end at stop codons. Is everybody good with that? All right. Okay, this is another version of the same picture that you've seen before. The difference is it's showing individual things. It's showing the monk-like scribe, the RNA polymerase. It's showing it bound to the promoter and reading to the terminator. Good? And what's spit out is the initial RNA transcript that we have to edit, the pre-messenger RNA. Okay, so this pre-messenger RNA has to be edited. We have to remove the introns from it, hook the exons together. They are allowed to exit the nucleus as messenger RNA. Are we good? So the messenger RNA is the recipe in that cartoon. It leaves the nucleus, leaves the capsule, goes out there and is read by the ribosome. It's read by the chef. Everybody good? All right, RNA is written in codons. Each codon is 3 nucleotides long and it tells you what amino acid to use. There's 64 different codons for the 20 common amino acids. We started a start codon, we ended an end codon. There's It should say we started start codon, we ended a stop codon. There's 1 start codon, 3 stop codons. Everybody good? Now, I'm going to tell you that what I wrote here, this is true for normal cells. There's a slight variation in what happens. Normal eukaryotic cells. There's a slight variation on what happens in the mitochondria. There's a slight variation on what happens in some bacteria. I'm not scared of those variations. I don't buy into this all, oh, but there are variations. That means there's no evolution. There's some people who like to go down that path. Not only am I not scared of those variations, I link to them on my website. Go look at them. Have fun. Okay. They're just variations. They're minor. But when we talk about the genetic code, I often tell you it is unambiguous, it is redundant, and it is mostly universal. Oh, remember I said I would link to those variations? There are the variations. Good. So when I say it's mostly universal, it's similar. So here we go. Unambiguous. What do I mean when I say it is unambiguous? What I mean is there is no question what it means. Give me a codon, any codon, Daniel. Just give me 3 nucleotides. Come on. Okay. UGU, perfect. So we come down to the U's, we go over to the G's, UGU. It's a cytosine. Everybody good? Okay, it's right here. UGU is right there. That is unambiguous. There is no question what it means. We know exactly what we're looking at. Do we agree? That's unambiguous. So if I say, what does it mean when we say that UGU is, what does it mean if we say that DNA is unambiguous? Don't get all dictionary English majory on me. Oh, it means we have no question about the meaning. Say, it means that we know exactly what a codon tells us to use for the amino acid. Be specific so I know that you understand. Does that make sense? Okay. Now, the other one I said is it's redundant. There's more than one way to say the color of Daniel's backpack. Or sorry, there's more than one way to say the color of your backpack, right? Red, scarlet, crimson. More than one way to say use proline. Got it? It's more than one codon. All right. Sorry, Jonathan. It took me a second.
My backpack's the blue one.
Oh, it's Brooke's backpack. It was behind you. I assumed it was closer to his desk. All right. Brooke, there's more than one way to say, now I understand the look on your face. There's more than one way to say, what is the color of Brooke's backpack? Or to answer the question, what is the color of Brooke's backpack? Good. Red, scarlet, crimson. There's more than one way to say, use proline. You could say CCU or CCC or CCA or CCG. That's what I mean when I say the genetic code is redundant. But the one that I love is the universal. And yeah, people get caught up. In fact, there's some minor variations. But the genetic code is so similar that I could take the gene that makes a firefly's butt glow green, and I can put it in a plant, and the plant knows how to make that protein. That's pretty universal. I can take a human gene and put it into a bacteria, and it knows what to do. That's how we get insulin. Does that make sense? Okay. That's how we get clotting proteins. That's how we get a lot of our medicines. All right, so you know the codon chart. You've seen the codon chart. You've used the codon chart before. Understand it. Don't just glance at it. Know that you read down for the first letter, you go over for the second, and then you could go with read down within the box for the third, but I usually just look at the box. Anyway, but you need to be able to use it. It's just like you had a periodic table on the test, you're going to have one of these on the test. Good? All right. So we know that we do transcription in the nucleus, we do translation out of the nucleus, that's fine. The codon will tell us what amino acid we're going to use. Ribosomes in the cartoon are the chef. They're what do this. They're mostly rRNA, but they have a little cap of protein, smaller subunit of protein. Everybody good? All right. There are 20 kinds of transfer RNA because there are 20 common amino acids. There are other amino acids, but the ones coded for in DNA, there are 20. It's shaped kind of like a clover leaf if you had it flattened out, but it folds up into this cool 3D shape. There are only two parts that I really care about. The anticodon that has to match the codon, and the amino acid that it carries at the other end. Good? So how does this work? Easy. Here we have a codon of RNA, messenger RNA, and here we have an anticodon of transfer RNA. If these two match, the ribosome says, well, yes, that's the amino acid I need. If they don't, it says, go away. Good? So the anticodon is renucleotides at the tip of the transfer RNA. It has to match the codon. If it does, that tells the ribosome who you take the amino acid from. The ribosome adds the amino acid to the growing chain. Actually, it takes the whole chain, breaks it off. Takes the whole chain, breaks it off, and moves it to the new amino acid. This continues up until you get to a stop codon. Then the messenger RNA, then the ribosome lets go of the messenger RNA, lets go of the protein, and the ribosome actually comes apart usually. Does that make sense? Everybody good? All right, here's a pretty picture of it happening. You can see that. Now, if you were a major, I would make you memorize that there are 3 positions in the ribosome, the A, the P, and the E, and we're not going to worry about that. Just understand that we have one that we've already read. We already decided, yes, we're going to use that methionine. That's a start codon. We have the next one come in. We're moving forward. What that does is you break off the old piece and put it on the front. And then the old one goes away. Then you move to the next codon and you wait for a match. And when you find the match, you break off the chain, put it on, and the old one, which is now empty, goes away. You do it over and over and over again until you get to the stop codon and the protein floats away. Are we good? You look really bored, Bryson. What can I do to excite you? No, you're good. All right, all right. So, would you agree that if you change the DNA, it might change the protein? Would you agree that the change might have no effect at all? Like say, oh, you change the DNA, so you change the RNA from this to this. Meh, who cares? It doesn't do anything, right? Because you're going to use the same amino acid. So that means you could have no effect, you could have a minor effect, or it could be something really big and bad. Mutations come in all flavors. But we have families or groups of amino acids that have the same properties, act the same way, and you don't have a lot of effect if you substitute between those. Good? What I'm saying here is all of these in this row here are hydrophobic. They hate water. Easy. All of these are hydrophilic. These are hydrophilic and positive. They love water and they have a positive charge. These love water and they have a negative charge. These love water and they have no charge. No net charge. A little part here might be charged, but the overall charge, non-existent. What do you think happens if we take a water-fearing one and we substitute it with a water-loving one? And water starts sticking to that protein, and that protein starts to react and go, and tries to get away. It's going to turn itself inside out to get away. Does that make sense? Or what do you think happens if we take a water-loving one on the outside of that protein, we substitute a water-hating one? That protein will turn itself around, it'll twist, it'll bend to try and protect the water-hating one from water. Then the protein changes shape. And if it changes shape, it does what? I'm looking for a D word? Denatures. And does it work anymore? No. So you guys are good with that, right? Now you do see this where it says this chart is on the test to be able to read it, right? So you know, don't memorize the chart. What you're going to do, both of these charts are going to be supplied, and you're going to have to be able to read them. There are examples similar to this. They're not the same ones. They're on the test. Hey, which of these mutations would cause the biggest change? If you're changing AAA to AAG, that doesn't do anything. Do you agree? because they both say use lysine. So AAA to AAG, nothing. Now, AAG to AGG, lysine to arginine. Well, there's a change. We know there's a change. The first thing we're looking at is, was there a change in these amino acids? But lysine, which is long, water-loving, and positive, or arginine, which is long, water-loving, and positive, do you think that's going to have a huge alteration in the protein? No. But AGG to AUG, change arginine, water-loving, to methionine, water-hating, that could cause problems. You've just substituted something from a different family. That mutation is a bigger deal. Are we clear? So there are mutations that alter just a little bit at one spot. We call those point mutations. And they're also called substitutions. The substitution is a point mutation. You substitute this for that. They might be a big deal, they might not. We just showed you examples of ones that weren't. We showed you examples of ones that were. There are bigger changes. Insertions and deletions usually cause what's called a frame shift. If you shift the reading frame, you shift what you're reading as this codon and this codon and this codon. You know, there's nothing in the RNA to tell you what's the first codon, what's the second codon. So if you start reading here and you get part of the first one, part of the second one, it's going to be messed up. It's going to be messed up from that point on. It doesn't just change one point. That's what a point mutation does. It changes everything. All right, so point mutation. Here you have the normal gene, and you make isoleucine. If you substitute a C for a T, suddenly, oh, we're making valine instead. But it's just one point. Now, is it always just one point? No, you could have a point mutation that created a stop codon, and you'd never get to proline. Does that make sense? But in general, point mutations have the chance to be not as big of a deal. Frame shift is going to mess up not just the one you're in, but the next one and the next one and the next one and the next one and the next. It's going to mess up all of them for the rest of the protein. Now, if you have a frame shifted into the protein, that won't be quite as horrible as if you have one at the beginning of the protein, because the one at the beginning would throw off the whole protein. If it was in the second to last codon, that's probably not as bad. But if you mess up your stop codon, then you don't stop, you read into other things, you got problems. Does this make sense? All right. Oh, are you fidgeting, trying to vacate the premises? Okay, that's fine.